Hello everyone. I recently gave this talk at the American Society of Pharmacognosy meeting, and I thought I would share it a bit more widely because it is a good introduction to what my lab is about and what we are currently doing. So if you are a prospective grad student or postdoc, this might be useful to you. Before we start though, I just have to briefly introduce the concept of natural products or secondary metabolites. These are small molecules made by living organisms, and I have long been fascinated by their intricate chemical structures, like the one shown here. These are generally difficult to make through synthetic chemistry, but they are also very often bioactive, and that has translated into many natural products becoming drugs used in medicine. But why are such intricate structures made in the first place? Well, the fundamental concept that is the absolute core of what we do is that secondary metabolites are products of evolution. Take, for example, this sessile marine tunicate called Ectinosidia turbinata. It is a soft-bodied invertebrate fixed to an attachment point in the sea. It can't really do much if something comes along and wants to eat it. However, it turns out that E. turbinata does have a form of defense, a secondary metabolite called ET743, that is a cytotoxic compound that alkylates DNA and also disrupts the activity of certain transcription factors. And this is important for us because it turns out that ET743 had a useful enough activity that it was developed into a drug marketed as Yondelis to treat soft tissue sarcoma and ovarian cancer. So if secondary metabolites are products of evolution, then where should we look for them? Well, the obvious answer is that we should find organisms that have a plausible reason to evolve some sort of bioactive small molecule. For instance, this tunicate is fixed in one place and is kind of soft and squishy. It's reasonable to think that it might be vulnerable to predators trying to eat it if it didn't evolve some form of chemical defense. And in nature, defense often means something that is not just unpleasant to eat, but is poisonous, so that things that eat the tunicate will be selected against. Of course, we know that in cancer, we often want to poison cancer cells just a little bit more than normal cells, so we can use a lot of compounds that are poisonous in nature to treat cancer. But evolution is not quite as simple as this picture makes out. As well as being a competition between all different organisms, sometimes it is advantageous to form alliances where both parties benefit. For example, a bacterium living inside a tunicate may be given a hospitable environment, can feed on metabolites made by the host, and it may not have to directly compete with other bacteria. What does the host get in return? Well, it seems like rather than evolving your own small molecule, it is often much more parsimonious to borrow the biosynthetic capabilities of microorganisms who evolve much faster. And in actual fact, David Sherman's group were able to show that ET743 is made by a bacterial symbiont of a tunicate, not by the tunicate itself. So perhaps what we should do is look for these types of symbiotic relationships for promising natural products. My group certainly thinks such systems are very interesting indeed, because if you find something like this, it may have taken millions of years to evolve, with the host essentially selecting for the most advantageous secondary metabolite it could get its hands on. But there are unfortunately plenty of challenges in studying these systems. What we know about these systems is that microbial symbionts all evolved from free-living ancestors, and free-living bacteria, of course, all have a certain number of genes that they need to live in their environment. This includes things like the biosynthetic pathways for amino acids, vitamins, and cofactors that it needs, and ways to respond to environmental conditions. But when a bacterium becomes persistently associated with a host, purifying selection pressure on a number of genes is reduced, perhaps because the host provides the products of those genes, and over time they become non-functional pseudogenes. Bacteria tends not to keep non-functional genetic material around for long, so over time those parts of the chromosome are deleted, making the genome as a whole a lot smaller. Additionally, because there are lots of very small populations of symbionts within individual hosts, they are vulnerable to extra gene loss through frequent population bottlenecks. Basically, any gene that is not absolutely necessary can be irreversibly lost, even things like DNA repair systems. Very old symbionts also lose things like genes necessary to maintain a regular rod shape, and they tend to look kind of blobby. The upshot of all of this is that the older a symbiosis is, the less likely it is that the symbiont is able to survive outside of the host or in the laboratory, and this process is irreversible. But on the other hand, the oldest defensive symbioses are probably the most interesting to us because they have been ecologically important for the longest. It's sort of a catch-22. The most interesting systems are also the hardest to detect and study. So how do we study these types of systems? Well, let's suppose that we have a beetle that contains a number of different types of bacterial symbionts within it, and none of these can be cultured. What you can do is extract DNA from the entire beetle, and you should get out DNA from all of the component species that are present in your sample. And we can then sequence all the DNA that we have extracted, and this should include both parts of the host beetle genome as well as fragments of the chromosomes from each of the resident microbial symbionts. However, here we have a series of problems. First, we never really know how many species our data comes from, and of course that is one thing that makes it difficult to separate the data into genome bins. The second big problem is frequently the uncultured species we are sequencing here are very unlike anything that has ever been sequenced before, and so we can't do things like use a closely related genome as an alignment reference. The final problem is that we are not sequencing just one genome here, but an unknown number, most likely much greater than 1. So that means that in order to get coverage of low abundant species, we have to sequence much more deeply than we would otherwise do. And that is more expensive, of course, but also you risk not seeing the rarest species at all if you don't estimate the right depth to sequence. And of course, there are various ways to separate genomes from metagenomes. This process is called binning. And actually, my lab developed our own pipeline to do this automatically, called Autometa. And if you would like to try it out, version 2.2 is out now, and here are the details. We have published a paper on version 1, and we are currently working on a paper for version 2. What I can share with you now is that Autometa 2 has much better performance compared to both Autometa 1 and other binning pipelines. This is measured by adjusted RAND index, which basically looks at whether contigs tend to be grouped with other contigs from the same genome or not, and it is the measure used by the CAMI challenge which evaluates binning programs. We can see that Autometa 1 actually didn't do great compared to the other pipelines, but we've overtaken them with Autometa 2. There is a range rather than one result, and multiple columns, because we tested a range of settings and parameters. These results are using the gold standard assembly or GSA from the CAMI2 challenge, derived from a marine metagenome and a metagenome with a lot of strain heterogeneity respectively. You can see that having lots of strain variants present continues to be challenging for all binning pipelines, and so there is some work still to do. But one thing to note here is that Autometa does its best to cluster contigs based on a few things: nucleotide composition, coverage, taxonomy of contigs, and the presence of universally conserved single copy marker genes. It does a good job, but often binning can be improved with some amount of manual polishing and curation. So we have developed an interactive web interface which allows users to refine Autometa results, called Automapper. We have already released the code for this, and we will be submitting the paper quite soon. We can see on this screenshot that there is a main chart in the middle. This is showing a graph where each point is a contig, and what we have done is use an embedding algorithm called Bundt Stochastic Neighbor Embedding, or BHTSNE, to convert the frequencies of 5 base pair nucleotide sequences into 2 dimensions. So points close together on this graph have similar nucleotide composition, and ones further apart are dissimilar. You can see that in this example there is one small red group of points that has been put into bin 0001, and the other points are all unbinned. We'll now replot those points on a bigger graph so that you can see what is going on. Now, in Autometa, we actually don't just use the BHTSNE coordinates for clustering, we use a third dimension, coverage. And as before, the red contigs were all binned together by Autometa. These data are actually from the metagenome of a marine tunicate, and the red bin turned out to be the genome of a symbiont in the phylum Bruchomicrobia that we called Candidatus didenratutatus Mandela. There are some pink points. These are a number of contigs which constitute a large trans-AT polyketide synthase, or PKS cluster, that we predict to make these polyketide compounds called mandelides. These are cytotoxic compounds which presumably protect the tunicate from predation. But the point is that these are much higher coverage than this symbiont genome, and so were not binned with it initially. But it turns out that if you manually align the paired end Illumina reads to both those sets of contigs, you can find connections between them that were not made by the assembler. And the reason it got confused was because there are multiple copies of this bisynthetic gene cluster, or BGC, in the genome, up to 7 for some regions. This assembly is also complicated by some deletions seen as links between distal regions of the BGC in some repeats. So even though we can't show this chromosome as a complete circle because we can't tell which version of each repeat goes in which order, this assembly is actually quite near complete. And looking further into this genome, we can tell that it is going through the process of genome reduction, and it has even lost some DNA repair pathways. So what that means is pretty astonishing. The BGC takes up over 25% of the coding capacity of the chromosome, and that means that it is incredibly important and is probably being heavily selected for. And it turns out that you can find other examples of this sort of thing with careful examination of metagenomic data. Here I'm showing a Forsythia sponge collected by my collaborators Amy Wright and Gojun Wang. This sponge is the source of another cytotoxic polyketide compound called lisonolide. If we look at the BH-Teasney graph of the shotgun metagenome on the right, you can see it is a lot more complicated than the previous tunicate example. But we could fairly easily find another trans-AT PKS that likely makes lisonolides. Now on the graph, the different colours represent different bins, but the question is, which one does this last BGC actually belong to? Well, in this recent paper, we propose that it belongs to another verrucomicrobial symbiont that we call Candidatus thymophili lisonolidus. Now, LAS is quite far away from that symbiont on this chart, and what that means is that it has very different nucleotide composition to the genome it is in. And what that likely means is it was recently acquired by horizontal gene transfer, and it hasn't yet had time to normalize its composition to its genome context. And we know that LAS is part of this genome because we see evidence of the LAS contig being joined to multiple T lisonolidus contigs, 3 on each end, and this means that there are 3 repeats of the BGC except for one small part on the left, which is only in 2 copies. Interestingly, this genome is not as reduced as the mandelolide example, but there is something, it seems, about verrucomicrobia that perhaps make them likely to duplicate BGCs. There was actually another example of a repeated trans-AT PKS in an uncultured verrucomicrobial symbiont published by Alison Murray and Bill Baker. Perhaps in all cases there is selection pressure to increase expression of the BGC through gene duplication. So here is another interesting example. This is a beetle called Lagria villosa, and it is a soybean pest found in Brazil. It lays its eggs in soil in these kind of clusters. Of course, soil is filled with pathogens, including a lot of fungi, and the eggs of this beetle are vulnerable to infection, which prevents them from developing. Fortunately though, the beetle has a chemical defense called lagriamide. This is a complex polyketide that is antifungal, and so it reduces the infection rate of eggs in the environment and thus increases beetle survival. My collaborators Martin Kautenpoth and Christian Hertweg isolated lagriamide in microgram quantities from about 50,000 eggs and determined the structure. But lagriamide might look a little familiar to some of you, and that is because it is very similar in structure to bistramide, a compound that was isolated from a marine tunicate. The only differences in structure are fairly minor at the ends of the polyketide chain. So that, in combination with the fact that these look a lot like bacterial polyketides, made us wonder who makes lagriamide and bistramide A, and whether it was likely to be a bacterial symbiont. Now, Martin Kautenpoth's group tried really hard to isolate the lagriamide-producing symbiont. One thing they noticed from 16S studies is that the eggs are consistently associated with multiple strains of Burkholderia gladioli, so they tried to isolate those. One of the strains they got was this one, LVSTA. And it turned out that LVSTA did make a number of compounds, even an antifungal one, but not lagriamide. Martin, of course, looked in the genome of this strain and couldn't find any BGC likely to make lagriamide. Also, upon further investigation, it was found that this particular strain was not always present on the eggs, so it seems that the antifungal this strain makes is not that important to the beetle and is not being selected for as strongly as lagriamide. So the question arises then, is the lagriamide producer an unculturable symbiont? And we can of course find this out through metagenomics. So we examined the shotgun metagenome from the eggs collected from Lagria villosa in Brazil, and here is a list of the genome bins we were able to separate. You can see this is quite complex, and also that there are many related Burkholderiaceae strains here. Note also that LVSTA is not one of them because it isn't always there. And here I'm showing the BGCs we found per bin, and I'm highlighting here the LGA pathway in Burkholderia species LVSTB because we think that is the cluster that makes lagriamide. You can see here that many of the strains present have BGCs, and some are quite large like the LGA PKS cluster. We of course already know that LVSTA has a lot of pathways, so I'll add that here for comparison. So it seems from this that even though lagreamide is consistently associated with Lagria villosa, it has a whole community of other strains that are more intermittently present, and a lot of them have BGCs that may act as options for chemical defense that could in the future become selected for, or perhaps they are already extremely environmental location specific. Another question this brought up in our minds was which of the many symbionts present have reduced genomes, because the ones that have been reduced may be more dependent on the host and might have been restricted to that host for longer in terms of evolutionary time. So to get to that question, we can look at the size of each bin, and once again we can use LVSTA as a closely related comparison. Remember though that our bins are in a number of different orders, and so perhaps it would be more accurate to compare each bin with its particular closest relative. So that graph looks quite different now because different bacteria have different size genomes. Let's reorder this then in descending order of percent size. It is kind of arbitrary, but we can consider bins less than about 65% of their closest relative's genome size to be small, but in metagenomics we are often not sure if we are missing parts of the assembly or if the bin we have really is that small. The question is whether these are reduced genomes or they are actually just incompletely assembled. Okay, so to get at this problem, we can look specifically at a set of genes which are found almost universally in bacteria and are known to be almost always there as single copies. By looking at which ones of this set are present, we can estimate the completeness of each bin. You can see already that there are some bins which appear more complete by this measure. However, this way of estimating completeness is complicated because we have found that reduced genomes can actually appear incomplete by this measure when we have actually assembled the entire chromosome. So to get around that problem, we also look at a reduced set of genes that are known to be present even in the most severely reduced genomes, except for perhaps organelles. The top of this graph then is complete in both single copy markers and core genes, so these are likely pretty complete bins. The bins I've highlighted here are incomplete by both single copy markers and core genes, and remember this is in the part of the graph where we had bins that were small compared to their closest relative, so these are likely incomplete rather than reduced. But only LVSTB is both small and complete by both single copy markers and core genes, so it's likely a reduced genome symbiont, not a part of a larger genome that hasn't been assembled. But what is LVSTB missing? Well, it turns out quite a lot. Here is a representation of all of the orthologous group of proteins we found in LVSTB and close relatives, and it is missing quite a lot of genes because it is very small and its relatives have quite large genomes. There is only a very small group of core genes it shares with its relatives. But perhaps a little surprisingly, it has a number of unique ortholog groups not found in its relatives. These might have been acquired by horizontal transfer or alternatively lost in all the other relatives. And it turns out that the lagreamide or LGA pathway is one such horizontal transfer. Here again we are seeing the plot that Autometa uses for binning, and LVSTB is very high coverage, almost 2000x. The lagreamide BGC is the same coverage as that, but is far away on the XY plane, meaning it has very different nucleotide composition. Now before in the other metagenomes we proved a connection by looking for paired end reads aligning across contig junctions or by looking at the assembly graph. That proved quite challenging here. However, we have found by single cell genomics that this BGC does co-localize with this genome. So what does this mean? The different nucleotide composition suggests that the pathway was recently horizontally transferred to the symbiont and it hasn't had time to normalize to the recipient genome. So here I'm showing a dot for each adjacent cluster of genes that seem like it was probably horizontally acquired, since it was more closely related to some other species than Burkholderia gladioli. And we also very crudely estimated the age of that acquisition by looking at nucleotide composition. I should stress here, this is a very approximate method, and you probably can't trust the absolute numbers, but the order should be roughly correct. Turns out the oldest acquisition was a few phage genes, and we are still not sure why they have hung around since then. Then the oldest acquisition we can find was the lagreamide BGC itself. Presumably after that, there was selection for the strain to become more heavily associated with the beetle and become more dependent on it. But the surprising thing is that we continue to receive horizontally transferred genes after that happens. With a lot of genome-reduced symbionts, this is not possible because they are genetically isolated from other bacteria, but of course we know that that is not the case here. There is lots of other strains present. The interesting thing is that the genome reduction was so selective for this particular strain only, and that the horizontal gene transfers seem to have not been enough to counteract the genome reduction. Interestingly, the genes that are required include transporters, perhaps to take up metabolites from the host that can no longer be made by the symbiont itself, and DNA repair components. So that is quite interesting because it suggests that some of these acquisitions may have compensated for gene losses. From looking at the rest of the genome, we know that it has lost a lot of DNA repair genes and primary metabolic pathways. So what I showed you up until now has been based on a single metagenome from some Lagria villosa eggs collected in Brazil. But it turns out that this whole genus of beetles has evolved special structures to hold bacterial symbionts and spread the mixture on the surface of their eggs as they are being laid. So that and the level of genome reduction in LVSDB suggests that the symbiosis has existed for a significant amount of time. But we want to know more about how this situation evolved, so we collected more samples. Firstly, we have an additional sample of L. villosa. Then we also have multiple samples from Europe, Japan, and Australia of multiple different Lagria species and one Echinolagria, which is a different genus that has the same structures. Now, the phylogeny of this group of beetles is not entirely settled, but when you sequence a metagenome, you usually are able to assemble the mitochondrial genome of the host at the same time and do phylogeny with that, and that is what we are seeing here. Now that gets kind of interesting when you compare it to the symbiont tree. Here we see that all these beetles have a reduced genome symbiont with the LGA BGC in green. There are also other things we assembled that are not reduced and don't have LGA in blue, and things that were isolated and grown in the lab from beetles that all of course don't have LGA and they're in purple. We also made a tree of the LGA genes shown here. Now one key point is that the beetle and symbiont tree are not the same. I'm showing one key difference here. This might indicate that the symbiont has not been strictly vertically transmitted from the common ancestor of these beetles. The second thing to notice is that the LGA tree is largely congruent with the symbiont tree. Now this is kind of strange. What it suggests is that there was one LGA acquisition, and that would likely be at the root of the symbiont clade, but it appears that a large number of extant strains subsequently lost it. However, it seems like there must have been some delay before genome reduction happened after LGA acquisition, because that is normally an irreversible process. So this tree seems to suggest that the different clades of green symbionts might have independently become reduced. This is kind of a strange conclusion. So right now we are in the process of getting long-read data in order to resolve the question of whether there is one LGA acquisition or not. If that is the case, we would expect it to be in the same genomic context in each strain. Okay, so the sorts of studies I've described to you tell us which compounds have been evolutionarily important, and so probably very interesting to us. But you may be wondering how we can get anything useful out of this. Well, I think the answer here is being able to leverage big data. And what I mean by this is that all uncultured symbionts had free-living ancestors, and also all these symbionts that had BGCs horizontally transferred to them got those BGCs from somewhere. So that means that we may be able to find relatives of symbiont BGCs in free-living bacteria, but they may very well be very distantly related, or the genes may be shuffled, or the pathway may be fragmented across contigs in a poorly assembled genome, something like that. So I'm sure a lot of you are familiar with the basic concepts of genome mining. There is, of course, a lot of DNA sequence information in public databases, and you can do things like this query here fairly easily, although it might take some work. So in other words, we're looking at the distribution of terpene synthases across all phyla and environments. The environments part might be a bit harder to access, but you can do it through NCBI if you look at the metadata and perhaps the linked PubMed entries for different genes. But a query like this second one might be a bit harder. So here we want to find all organisms from sponges, so including bacterial strains, that have PKS genes near a halogenase and at least one antibiotic resistance gene, that are also not annotated by anti-smash and only in the cases where the strain is available from ATCC. So this is doable, of course. All the data is out there on the internet, but you have to know where to look, and you also have to integrate data from different sources. So of course, as a PI, I am maybe out of touch and I don't know how long things take, but I would probably expect a grad student to do this in a couple of days. But actually there is a type of data structure that should be able to resolve this in a couple of minutes, and that is a graph database. Essentially, this is a sort of database based on connections between different types of data rather than set tables. So we set out to make a global natural product and biosynthetic gene cluster graph database to facilitate all sorts of semantic gene mining. And of course, a backbone of this is information from NCBI. So from NCBI nucleotide records, we can connect to the assembly record and also to the derived protein records. And from assemblies, we can get taxonomic information. We can use the information from MP Atlas to connect specific strains to specific compounds. Of course, proteins are annotated a variety of ways from different sources. We have the PFAM database with HMM models for different protein domains, AMR finder for antibiotic resistance. There is also the MyBig database of annotated BGCs, and we can also run anti-smash on the database to get a global idea of BGCs. And of course, the entries in MyBig are also connected to compounds, and it also stores information on the activity of compounds, so we can link those pieces of information. We also know which proteins are similar to each other, and we pre-calculate clustering at the 90%, 70%, and 50% similarity levels. Likewise, we know what compounds are similar because NP Atlas has Tanimoto scores, which describe how similar compound structures are. So we can draw connections between BGCs that make similar compounds. So we wrote a pipeline to basically make such a database called SocialG. Right now, a lot of the code is public if you want to have a look, but we haven't yet released the global graph database itself. That will come out when the publication is accepted. So actually, this was the brainchild of my postdoc, Chase Clark, who basically single-handedly created SocialG. And this was quite challenging, actually. He only just got it to scale to the full-size database with all of NCBI on it. But now we have an automated process so that we can periodically update it automatically as those public databases grow. So I'm just going to go through a few initial examples, but really we are still exploring what can be done with this. So in the first example, we want to find every anti-smash BGC in all of RefSeq that has more than or equal to 70% of its proteins with sequence similarity to a BGC in MyBig. So what we are looking at here is actually a giant network plotted in Cytoscape. It is just that the nodes and connections are so small that you cannot see them. So to make this network, we started with 2,502 BGCs from MyBig, and we pre-ran anti-smash 6 on RefSeq, which has about 300,000 genomes, giving almost 2 million BGCs. And what we got out of that was a network with connections to 1,877 MyBig BGCs from a little over 300,000 anti-smash BGCs. And in the network, the nodes are colored by taxonomic order. And immediately you can visualize the structure of the network. You can see that there are large clusters of BGCs that are highly conserved in an order, and there are also ones which seem to have more wide distribution. The other interesting part of this is that there are quite a number of singletons in MyBig, but a much greater number in NCBI as a whole. And what I think this means is that we are very far from exploring all of the biodiversity that is out there, because I think it's probably unlikely that there are any BGCs that are completely unique to one strain in the entire world. And just to show you the power of graph databases, this entire network was constructed and returned from the database in just 2.4 minutes. And once we have a network in hand, we can further explore the data. For instance, here I am coloring the network by whether a strain is available in the ATCC strain collection. Green means yes and grey means no. So this sort of thing would allow us to easily find a strain that carries a similar BGC to the one that you are interested in, if it exists in ATCC, of course. Another question, can we use this database to find BGCs not annotated by anti-smash, or are we just restricted to our anti-smash results? Well, here is an example. On the left is a BGC annotated by anti-smash. That is the blue circle, and the component proteins are the orange circles immediately connected to that. And we can of course connect that BGC to a species and a compound. Now, like one would normally do, we can search the whole network for BGCs that are similar to the left one and have its protein components. And one thing to remember is that this is a whole large graph, and so the target BGC proteins don't have to be within a certain percentage identity to the query. They can instead be multiple jumps from them, with intervening proteins between them. So in this example, the target BGC is not within 50% identity of the query, but we can get there through intermediate proteins. And this new BGC we found actually is not annotated by anti-smash, and so we can use this to systematically find such biosynthetic dark matter. Another important point here is that we are just looking for the presence of homologous proteins. They don't actually have to be in the same order, and we might actually be able to find fragmented pathways in this way. And just to show you another quick example, Chase made a huge network with the whole of MyBig. This is just a small part of it. And you can see that even though we have just clustered this based on protein similarity, there is also a certain amount of structural clustering. So what that means is that you can use this to systematically look for BGCs likely to make structures similar to the search query, and perhaps find surprising connections. So as I said, we're still exploring what we can do with this pipeline. One of the things we hope to do is to be able to find distant relatives of Symbiont BGCs in free-living strains. Because up until now, metagenomics has been somewhat of an observational science, we hope that through these efforts we can move it into a place where we are getting more direct access to the compounds we use metagenomics to investigate. Okay, so that brings me to the end of the talk, and it just remains for me to thank my lab and collaborators, whose names are all up on this slide. And of course, we couldn't do any of this without our funders either. And I would also like to thank all of you for watching this video.